Hello, everyone. I know you are really tired, but I'm going to make it really short and just an overview of what we are doing in AI for Soil Health project. You see many co-authors, and because we are involved in different pilot sites and we are all working together for the project. But of course, why we are here, all of us, because the importance of the soil, and we know that soil is giving us many natural and ecosystem services. But growing population and mechanization put a lot of pressure on the soil. But when they want to also do like observation, the only focus is only chemical and physical properties. But also, as Tom mentioned in the discussion panel, that we have a lake of biological functions that we need to assess them as well. We only have 600 data points for biological function across the EU. But to do the biological function, we also need to have a set of indicators to define like soil health indicators. We all know the importance of the soil biology, but there are really less studies that they are assessing those. We know the importance of soil biology in microorganisms, nutrient cycling, and water retention, and many, many other properties. But we need to define and set up soil health indicators that can be used for all farmers, advisors, should be easy to use. And most of these methods, except eDNA, it's really easy to use, but eDNA, it's a bit more complicated because we need to send the labs to the samples to the special laboratories. But this method that I'm going to talk, it's about eDNA, enzymatic activity rate, fungi to bacteria ratio, and water infiltration capacity. First is about eDNA. If I want to define eDNA, it's any genetic material coming from the soil, air, or water. We call it environmental DNA. And the method to find the eDNA of the soil is called metabarcoding, and we need to estimate the biodiversity and also the composition of different soil taxa. For example, the fungi, the bacteria that they have, but also, I guess my slides, a bit, yeah. Yeah, I wrote something, but it's not here. That dominated biological function also can be predicted. Like the steps of nitrogen cycle also can be predicted from eDNA results. And the methodology is quite simple for us because we just send it to the lab. But what we do here is we just add the fresh soil in a small tube, and then we add the DNA and RNA shield, and we just send it to the special laboratories, and then they just do the DNA extraction, amplification, sequencing, clustering, and taxonomic assignment, and health assessment as well. For example, here, one of the results that we already have for different countries, so I have to say that eDNA results, we have the result in two categories. So first is the richness, and the second is the diversity. Richness is about how many different types of fungi we have. And you can see here Denmark, Sweden, and Croatia, and Finland. We see that the richness of fungi in these countries are quite similar in Denmark, Sweden, and Finland, but not the same as Croatia. But the diversity also is the same. We can see that the soil samples, each of these points represent one sample. We can see that the points in Sweden, Finland, and Denmark, they have a bit of overlap, but in Croatia, they are a bit different. The other methods that we are using, it's soil enzymatic activity. Enzymes are releasing in the soils by microorganisms living in the soils. And we can measure their enzymatic activity. And of course, we know that enzymes are really important in soil processes, such as nutrient cycling, organic matter decomposition, and also decomposition of soil pollutants. What we are doing here, it's a startup company from Switzerland, and they are also here. If you have any detailed question from them, they are sitting there, Jasmine, and they also have a poster presentation in the afternoon. So I'm just say what we are doing. We collect the fresh samples and we put it in a cuvette like this. And they have a device, they call it Bob. And they add the fresh soil here, and we add a reaction gel and onto the soil, and then we put it into the Bob, into the readers. And then we will have the enzymatic activity in 5 different categories. But there are benefits of these methods because it's easy to use, and also it has the minimum disturbance of the soil. And the error is really, I mean, is there are error reduction also, and also the method is good because it can be adaptable for a specific soil properties. The other method that we are doing is macrobiometer that gives us the bacteria, fungi to bacteria ratio. We know the higher microbial, we know the higher fungi to bacteria ratio, the more nutrients available into the soil. And this method is quite easy because you just need to have the portable kits, and the farmers can go to the field and they can check the healthiness of their soil by just doing the test. The test is only 20 minutes, really cost-effective method. And they can check the healthiness of their soil, and they can see if, for example, the management practices that they are working is really working or not. And the methodology is quite easy because they just collect the samples in those tubes, and then you add the extraction powder and just you let it be for like 10 minutes, and you add 3 drops of water into this small sheet, like a small, and you take a photo with your smart app, and then you have the results of fungi to bacteria ratio here. Yeah, the farmers can go easily with these portable kits, and then they do their measurements. But the other methods that Peter also was talking about is infiltration rate that gives us infiltration capacity and hydraulic properties of the soil. We can know about the pondings and erosion. But what we are doing in the AI for Soil Health project is we just have a small, really simple team, and then we just add the waters. There are different methods to do the infiltration capacity, but this method needs only few liters of water. And then we just add the water into this tin, and we record the time, and we repeat this process until we get a constant rate, and then we will have the infiltration rates. Yeah, but all of these methods are for what we are doing. It's about for the project AI for Soil Health that you probably heard it a lot from the morning. And we collect the soil samples from different locations. We have been in Sweden, Finland, Denmark, Greece, Croatia, and now after this conference in Netherlands. And we're gonna find this the correlation or analyze between these indicators. But are we going to forget about the wet chemistry as we are always doing? Of course not. We're going to have the biological function, we're going to have the wet chemistry analysis, and also a spectroscopy, because we want to see how these results are correlated. Or but also one thing that is important for us is to check their robustness and also their interdependence. For example, if the farmers doesn't have any access to any of those methods, can they just go to the field and just do the microbiometer, for example, the measurement, and then they still can have a status of their healthiness of soil or not? So the hypothesis is that we imagine that there is a significant positive correlation between these novel indicators that I explained. For example, eDNA richness is correlated to enzymatic activity, which is in turn correlated to infiltration capacity, and all of them probably will have the highest score of carbon properties as well. Yeah, and with that, I would like to thank you.